This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 165, recorded January 5th, 2012. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. And this is the first episode for 2012. Joining me today on this momentous occasion right here in the TWiV studio, Dickson Despommier.
Hello, Vincent.
How have you been, Dickson?
I've been well, thanks.
Well?
Yeah, perfect.
How's
I
the weather?
Great.
How's the weather where you are?
Well, it's the same as where you are, actually. The weather has been variable, actually. It started out really mild over the holidays, and it's now cold. It's absolutely cold out there. In fact, the chill factor in the Catskills about 2 days ago went down to minus 30 degrees.
Let's see, I got the temperature
That's
here
pretty cold.
For Gainesville, but New
you
York
guys
City,
had a killing frost down there, at least yesterday.
It's 4 degrees here in
Yeah,
New York City.
yeah.
So let's see what it is down in north central Florida, where we will find Rich Condit.
Hi, fellas.
Hey there.
I'm all ready for you.
Good.
It is 293.15 Kelvin.
Oh, Kelvin. It's got Kelvin now.
It is 527.67 Rankine.
Ooh.
66 Newton. I didn't even know these existed.
Newtons.
Minus 120 Delisle. 20 degrees Celsius and 68 Fahrenheit.
Ooh.
Interesting. 20 degrees C. That's the one I recognize.
But you guys had some killing frost down there recently, didn't you?
We did. Night before last, it was record low, 21 at the airport.
Exactly. So what crops would you be losing under those conditions? I mean, what's growing at this time of the year in Florida?
I don't know enough about that.
Oh, okay.
They're always paranoid about the oranges.
No, they are. That's
okay.
True. That's true.
And so the fruit trees are always an issue.
All right. Vince is raising his hands up, and he wants us to move on.
I just want to bring in our other member of the show from Western Massachusetts, Alan Dove.
Tomatoes. They would lose the tomatoes.
They would lose the tomatoes.
And yes, it's good to be here.
See, I wanted to bring him into the conversation because he's a good guy. He doesn't say anything till he's introduced.
Plus he knows stuff like tomatoes.
That's true. This is all true.
Hey, Rich, you're fading out.
Oh, no.
Oh,
really?
Your thing is on automatic or manual?
No, it can't be. Let me check.
Don't you think he's fading out, Dickson?
He sounds a little distant.
He is distant. He's a distant guy.
No, he's right up front.
I'm automatic. I can crank it up some.
Yeah, crank it up a bit. Yeah, it's good.
Is that good?
That's much better. Much better.
Excellent.
Leave it there, okay?
All right.
All right, today, the first show of 2012. I got to get used to 2012. That kind of slips off the tongue. Better than 2011. It's the same. I don't know. Hey, how are you, Alan? You okay?
Doing okay. Yeah, Rich just stole my joke. I was going to do the different temperature scales, too.
You were?
No!
Yeah.
Really?
You guys both thought of it independently?
No, I actually, right before I brought up Skype, I looked up conversion factors for Rankine, because I didn't remember, and I was just noticing that there were several other scales that I'd never heard of.
Yeah, I was reading off the Convert app on my iPhone.
Okay.
Oh, my.
Nice. All right, today we have an all-email episode. Your questions answered by us, or at least we try. But here they are. We have a bunch. We've got a bunch. Let's get through every one of them.
Ooh, could be here for a while.
Yeah.
First one is from Justin, who writes, are you, Alan Dove, and Professor Racaniello, saying you think Mikovits and or others on the Lombardi paper lied about the results or blinding? I think circumspection is a natural human reaction to the allegations of theft that have been made against Dr. Mikovits. My impression is that she was at least out of line, maybe worse, but I think we need to wait for all the evidence in those cases to come out before we make final conclusions. All right, let's pause and answer that. Alan Dove, I have not said that anybody was lying, and I will not say that anybody was lying until it's incontrovertibly proven, which in science usually means that the person comes forward and says that they were lying, or somebody comes forward and provides conclusive evidence that they were. We distinctly did not say that.
We did not wave the fraud flag, but I will say that, I mean, I've seen a lot of sketchiness coming from the Mikovits et al. Camp, and it comes in several flavors. There was the whole setting up a commercial assay for a virus that hadn't been proven to cause any disease, and advocating that people take toxic antiretrovirals for that virus, and going on a speaking tour around the world where people made talks at woo-woo conferences, talking about vaccines and autism and that sort of thing. And, there's a certain amount of that you lie down with dogs, you get up with fleas, I think.
We were just discussing the figure in that paper, which had been mislabeled. And so we're trying to figure out why that happened, because we don't have an explanation. And at best, it is sloppy science.
Yes.
If you mislabel your figure, it's sloppy. And for a science paper, you really shouldn't do it. Everybody makes mistakes, but when you make a mistake, please tell us what you did. Say, I messed up, and that hasn't happened.
And when the mistake is that consequential.
Yeah.
It really, this was not just somebody forgot to dot an I. This is really, it guts confidence in all the conclusions.
All right.
If you look at the legends for those two figures, they describe two completely different experiments.
Yes.
Okay. To me, mislabeling is not a satisfactory explanation for what went on there. I looked at that as carefully as I could.
Yeah. So I'd like to know the answer. I don't know.
Parasitologists would call that odoriferous.
Yes.
Something.
So that's what we didn't accuse anyone. You may have read it into what we said, but we would like to know what happened. But I think it's sloppy at best, and that's not good science. All right, moving on. And this rigorous study by Lipkin won't be believed by you say, if the results confirm the Lombardi paper. The burden is on them, not the other less rigorous studies. What I said, now moving to me now, I hate to break this up, but I think we should answer these one at a time. I said that if Ian Lipkin finds some viruses, they will have to be confirmed by others. We're not going to just say, Ian Lipkin found this, let's make a vaccine against this virus. Others will have to confirm, just as we said the Lombardi et al. Had to be confirmed.
And I said the burden is on them. And what I meant by that is, if you make an extraordinary claim that flies in the face of, I wouldn't call them less rigorous studies, I would call them a mountain of other studies that say that XMRV is not causing any human disease, and that the retroviruses are not correlating with CFS. If you're going to make a claim like that against all the other evidence that's already out there, the burden is on you to explain how you got that result. And the analogy I would draw is, if you make a claim that you saw neutrinos moving faster than the speed of light, which would overturn special relativity. Now, bear in mind, we've built an entire infrastructure on special relativity. My GPS works. I was just using it in my car today. If special relativity were wrong, it wouldn't. So it's not quite that far along with XMRV, but we're pretty close to that point where you'd have to look at it and say, how can all the rest of this be wrong based on one finding? So there would be an enormous burden of proof to say, you got to prove you didn't screw anything up, because that's going to be the first assumption.
All right, moving on with Justin. You guys do make some good points. There are multiple lines of evidence against XMRV infecting humans in vivo. However, your assumptions and conclusions about the pro-human gamma retrovirus scientists seem to me to be biased once again as compared to your conclusions about the anti-HGRV scientists, and your silence on the outright fraudulent scientists involved in XMRV and in ME science in general, e.g. CDC, Wesley School, including McClure.
Now who's making fraud claims?
I have no evidence that there are gamma retroviruses that infect humans. Does anyone else?
Right. I agree. I have no evidence, and that's where my sort of expertise on this whole thing starts and ends. I'm a virologist. This XMRV stuff came up. We've evaluated that story as carefully as we can. There's no other credible evidence that I can see that human retroviruses cause this disease. Period. End of story.
So
end of that story. Yes, there's a lot more on ME, but end of the virology story for now.
Yeah, for now, it's fine. Who knows what will come out.
Right.
And I see no evidence of fraud by these other characters who were just mentioned.
Mm-hmm. Correct. I don't
that's accusing the CDC and McClure and Wesley of fraud. That's a very, very serious charge, and I would expect some very, very serious evidence, and it doesn't exist.
Right.
They may be sloppy too at times.
Yes, and
not necessarily fraudulent.
Conclusions that you don't like are not the same as fraud.
Right.
Okay. So yeah, and research priorities that you don't like are not the same as fraud. There was some financial irregularity that occurred at the CDC that was investigated, and that investigation is a matter of public record. And at no point, as far as I know, was that ever called financial fraud, which would not even be scientific fraud. I believe that was accounted for as mismanagement, which is certainly not good, but it's not the same. It's not the same critter.
Moving on, there is some circumstantial evidence of potential sketchiness, R.E. Mikovits, and you have no problem assuming the worst, which is a normal human reaction. But why the double standard when it comes to the other side, the proven frauds who wage war on ME science? I have asked this numerous times and don't get an answer.
See above.
Yeah, Alan just addressed that. And he writes, I appreciate that you published David Tuller's piece on CDC, but that's all you've done. In this podcast, you mentioned that he wrote the piece, but you didn't mention that it's another documentation of the fake science done on ME by CDC. I can't think of another way to explain it but bias.
This is a virology podcast. It's not an ME podcast. Okay? We address the virology.
And it's not a CDC bashing podcast.
No.
Again, you can certainly question and disagree with and vehemently disagree with the research priorities of federal agencies or whoever. I'm all about that. That's fine. But to say that it's fake or dishonest or fraud just because you don't like those research priorities, that's not cool. And yes, there was a bit of a scandal. I guess it was in the mid-'90s with the finances at the CDC and how the CFS budget was being allocated. But as I say, that was investigated. That investigation is all a matter of public record, and there it is.
And by the way, I finally read Tuller's piece completely in response, prompted by Justin's letter, actually, and it's very good.
Yes.
So anybody who's interested in all of that beyond the virology should have a look at that. It's very good.
Thank you for your letter. Apparently you don't like what we do, but we try to be unbiased and evaluate the virology. And in this case, there have been a lot of issues, and one of them was a sloppy figure among others, and we pointed out. But if other studies have problems, we will point that out as well, but I doubt anyone would notice because none of them are as highly charged as this one. Rich, would you like to read Suzanne's email?
Sure. Suzanne writes, I believe the main problem some conservatives have with Gardasil is that the abstinence-only sex education programs in a lot of areas focus on, quote, sex will kill you, unquote, type messages. HPV is one of the pillars of their argument. You can't say, don't have sex at all. You're likely to catch this very prevalent virus that can cause cancer if your students have been vaccinated against the main cancer-causing strains. Good point.
It's kind of scary. It's kind of scary that they would do this.
Well, but
yes,
that's what happens.
Which makes the argument even stupider that people have against Gardasil. If that's your objection, then you have no objection. So, but thank you for filling us in on that, Suzanne.
Yeah,
I have not been in touch with where that argument came from.
That's very clearly stated. I think now I'm contradicting myself here because this is not virology. But my understanding is that telling people to not have sex doesn't work.
Right. That actually is not an effective strategy.
Has a reverse effect, actually.
Did you know personally, Dickson?
Well, I mean, it seems to be the epidemiological truth of the matter. You tell somebody not to touch wet paint, and the next thing you know, they've got paint all over themselves.
Right, and the observed finding on the on these particular abstinence-only sex education programs, which is, it's an education program defined by censoring particular information.
Yep.
And the evidence on it is that it doesn't work. That you do better by teaching kids what's called abstinence plus.
This is back to our transparency conversation, right?
Yep.
Somebody wrote to me the other day, if you tell people not to work on H5N1, that's going to make them work on it.
That's true.
Yes.
No, it'll be the impetus for it. That's right.
That's right.
No question.
Rich Dickson, I think you wanted to read Michael's, didn't you?
I want to read something. Come on, you guys are always reading these things. I'm just sitting here.
You get your chance.
Absorbing. So I will read this one. Michael writes, Great Men of TWiV.
That's why you wanted to read
Yeah,
absolutely. I'm a new virology/immunology postdoc. I am very glad your podcast exists to remind me of all the virus out there that I don't personally work on. Before I started listening, I'd forgotten all the details of everything except herpes viruses and pox viruses. You have a great camaraderie. No matter how interesting the subject, I wouldn't be able to listen if you sounded boring. For some reason, I was surprised to see that Rich Condit does not have a beard.
That's the second person. I guess I have a bearded voice.
Come on.
You do.
Rich, it's not too late. I'd like to know what you think of this paper.
I did that for 35 years.
Oh, okay. I'd like to know what you think of this paper. http//
No, you don't have to read that.
No, the other one. Okay. Manley et al., 2011. Human cytomegalovirus escapes a naturally occurring neutralizing antibody by incorporating it into assembling virions.
Virions.
Okay,
This is why we don't have you read letters.
Probably right.
Oh, come on, he's doing okay.
Cell host and microbe 10.197.
It's called a colon.
Stop. One of the
parasitologist should know what a colon is.
Hey, come on. Okay, somebody else want to take it up here?
No, we want you to read it.
Okay, one of the senior authors, Teresa Compton, gave a talk in our department recently, and she made reference to some interesting findings that she wasn't allowed to talk about. This seems like it is probably that finding. The idea of a virus incorporating an antibody into infectious particles is weird. Although the authors say, quotes, illustrates a general mechanism of viral antibody escape, they don't mention this happening with any other antibodies against CMV, cytomegalovirus, glycoprotein H, or any of the other CMV protein. He meant proteins, I think. Do you think this is a freak occurrence? Since antibody clones are unique to each person, I'd be surprised if a virus can evolve resistance against a single antibody clone, especially a herpes virus, which does not exactly have a high mutation rate. Best wishes, Michael.
Stop.
I did.
Okay. I don't want you to read his
No, I'm not going to. I'm just stopping. Michael.
Cut it out.
That's right.
This is a really cool paper.
It is.
Yeah.
I hadn't seen it, but it's published in September. This would have been a perfect one for an episode.
Yeah.
Well, it's not too late. Again,
but basically,
it's actually kind of the dengue thing used to advantage. Deliberately.
Yeah.
Right.
It's called sleeping with the enemy.
So they have a monoclonal against CMV, which is was tested in people to protect against infection, but it doesn't work very well. And so they found here by passing the virus in the presence of the antibody that the virus doesn't get neutralized anymore, but it's non-genetic. There are no mutations in the virus that make it escape the antibody, because you could say, well, the virus mutates, right, and the antibody can't bind anymore.
Exactly.
But what the virus does is the antibody gets taken into cells, and it gets put into the newly assembled viruses. It binds to the outer glycoproteins, I guess.
It actually binds to its target. Is that right?
Yeah.
Glycoprotein H.
Doing what it's supposed to do, but it doesn't do what it's supposed to do. Is that the idea?
Right. And then the virus uses the FC domain of the antibody to infect the next cell.
Yeah, and it goes in by a different entry mechanism.
That's mad.
And so normally CMV gets in by a different route, fuses at the surface. But with the FC binding, it gets in by endocytosis.
It's like stealing a cop car to go rob a bank.
That's exactly. Weird.
Really? Then inviting the cops in to drive you home.
I'm just thinking the virus is saying, oh, yeah?
That's right.
So the one thing amazing in this paper, so they took these variants, they sequenced the whole genome, and they found a few mutations, and then they passed the virus without the antibody, so it became sensitive again. They sequenced it again, and the mutations are still there. So they had to go through an enormous amount of work to show that this is a non-genetic phenomenon. It's simply antibody bound to the virus.
Wow.
It's just really interesting that the antibody gets into the cell, and then in the cell gets put on the virus.
Right. Now, this is a monoclonal antibody, so the part of the question about how it would vary from person to person, antibody responses would vary from person to person. It just so happens that this particular monoclonal antibody against this particular antigen was initially thought to be a potential treatment for CMV, and then it flunked. And then this is subsequently looking into why it might not have worked, and this is what they find.
So if you gave this as a curative, and the host responded by making anti-idiotype clonal antibody.
No.
Wouldn't that then interfere with this reaction?
It could.
I see. You're talking about a therapy.
Yeah.
Could, yeah.
I mean, because we're going to make anti-type antibodies when you give antibodies a therapy, right?
Yeah, then the virus is still there, and it's going to infect you.
Yeah, exactly.
Right, and apparently this thing was tried.
Yeah, doesn't work.
And it didn't work well.
Whether this is a generally occurring thing, who knows? Nobody's looked. I think now that we see it can happen, people will look.
Yeah.
Maybe it doesn't work with antibodies against other proteins. Who knows? Maybe this particular one it works for this. But these are good questions. We just don't know.
Yeah, but it seems to me that it very well could be a general mechanism for CMV immune evasion.
Yeah.
So why wasn't she allowed to talk about this when she gave her talk?
Yeah, she works at a company. But this is weird that she even said, I have something I can't talk about. That's ridiculous, okay? Now, maybe he's recalling it secondhand and it's different, but if you can't talk about something, don't even mention it.
That's true.
Yeah.
It's not fair.
Exactly.
Anyway, that's pretty cool, Michael. I bet, as Rich says, it's probably a CMV, a broad mechanism, and whether it works in other viruses is a really good question.
Yeah.
Alan, you're next.
So Chen writes, for viruses like CMV, here we go again, cytomegalovirus, HBV or HIV, those which are characterized by latent infection, what factor controls its reactivation? What determines the priority of its genome-encoded proteins being expressed, and how can the virus be detected as early as possible? Take CMV, for example. Most people are naturally infected with it. However, for immunocompromised people, CMV can reactivate from a latent state and cause great impairment to the host. As those people have a deficient immune system, their health relies on drug treatment combating the CMV when it reactivates. In such cases, detection of CMV as early as possible is the critical issue in ensuring the people's safety. Sincerely looking to your reply, forward to your reply and any advice. Best regards from Chen, who is in Beijing, China.
So
I asked Saul Silverstein.
Yes,
I'm not.
We have a response from Saul Silverstein.
Yeah.
Who is the expert on this. No one knows what controls the switch from latency to virulence. More likely than not, it's a manifestation of the host and the site of infection. Changes in expression of host genes or activation of heat shock proteins may be the most likely explanation for release from latency. And that would apply to herpes virus as as far as what's involved with the other viruses reactivating. I think they're possibly even less understood.
Yeah, the herpes viruses, CMV and others, we're talking about a different kind of latency than in HBV and HIV,
right,
where there really does seem to be an on-off thing with respect to gene expression. I think of HIV and HBV as more like persistent chronic infections, where there's a little bit going on all the time, and it's whether or not you see active disease has to do more with your immune system. But with the herpes viruses, gene expression actually turns on and off, and we don't know why that is. Another thing I see mentioned quite a lot is NF-kappa B, but you can almost answer any regulatory question by saying NF-kappa B.
Yeah, if you remember when we had Kathy Collins on, she talked about the cells latently infected with HIV. These are these long-lived, some kind of stem cell. And we don't really know what turns the virus on. I mean, she said, well, when the cells get activated and differentiate, some transcription factors are probably made that turn the virus on, but they don't really know. And even less so for HPV. That's correct.
These are tough questions to address because we don't have great models for reactivation for most of these viruses.
Yes.
Well, there's even an analogy here to another kind of infection in malaria that has a latent infection in the liver. No one understands what makes this come out every now and then, and up till 5 years after you've acquired Plasmodium vivax or ovale. So latency per se is a very poorly understood phenomenon.
All right, the next one is from Jason. Hey, guys, I thought of a cool idea. I think it was Scientific American that used to do a historical look back at what was going on in science 50, 100, and 150 years ago. I reckon it would be great to pick a random virology viral diseases article each week or month from 50 and 100 years ago and give a quick 30-second outline on what was going on, just to give us a bit of perspective on how far we have come. I like that.
Well,
I like that idea,
Didn't we do that with Peyton Rouse's paper?
Well, it wasn't a quick 30-second.
No, it was a whole show. That's true.
The only problem I can foresee with this is I don't know that we're capable of doing a quick 30-second outline.
No, we could never do it.
You could read the title in 30 seconds.
We would end up talking the whole episode about the 150-year-old.
Well, we could make a rule. We could say one person does it and nobody else says anything. 30 seconds. Right. We have to give it a cool name.
Start a shot clock or something, and I'll have to post
it. That's a good idea. I like it.
Yeah,
it is.
But we come up with a name of it. It'll be a new section, like 30 seconds. This was 30 seconds. I don't know. This was once. Whatever. We'll figure it out. Give us a good name. He continues. Keep up the good work, guys. You keep me company on the long drives to and from the lab each day. Actually, I have all 3 podcasts, TWiV, TWIP, and TWIM, on repeat in my car, so I must have listened to each episode half a dozen times each.
Holy cow.
I'm sorry about that.
It's got a long commute.
I'm usually paying most of my attention to the traffic, so I only absorb about 10% of what you're saying, which means I must be just over halfway to hearing everything in the podcast by now. All right, that's a good way to look at it. Aren't you tired of our voices?
Right.
Guess not. Anyway, he's from Australia, I guess.
Yeah.
It's pretty lonely there, right?
Well, no.
North Melbourne.
Melbourne is a lively cosmopolitan city. I spent 6 months there, and it's a great place.
Anyway, g'day from down under. The temperature is about 27 degrees centigrade, 25% humidity, and a light breeze. Definitely a day for the beach. Not here.
No.
P.S. I found the attached article yesterday. I thought you might find it interesting. You need a whale of a supercomputer to run a simulation like that. Makes my little poliovirus virion simulations seem like minnows. So he sent a paper called, it's in the Chinese Science Bulletin, Application of the Mole 8.5 supercomputer probing the whole influenza virion at the atomic level.
Wow.
Cool.
Wow.
They did reconstructions of the virion, dynamic simulations, and that's the end of my understanding.
Yeah, exactly.
Yeah, I read about a line of this and said, "Oh, okay." Wow.
So the results begin with, starting from the predefined structure, the virus experiences a significant change to obtain a stable structure. And then they show a couple of graphs. So, Jason, maybe you could explain this to us. Not sure what they're doing, but I take your word that it takes a lot of computational power. What is 8.5, mole 8.5? Number of calculations per second? Or?
I don't know. I thought it was a computer. I don't know.
Yeah, I hear these things spoken of in teraflops or petaflops or something, but I don't know. Maybe there's...
So, Jason, look, give us a 30-second summary of what this means. Yes. How's that? No more. All right, thank you.
Hey, how about yesteryear in virology, YIV?
YIV,
yesteryear in virology. Yeah, we do that maybe at the outset of each show. 30 seconds, no more.
And if we go to video, we'll do that part in black and white.
with a scratchy image, right?
Put on a top hat.
Good. All right, the next is Rich. I'm keeping a list here.
Uh-huh.
Okay.
Because I usually forget.
So where am I?
Peter.
Peter writes, Dear Vincent, Rich, Alan, and Dickson, as a fairly recent listener to TWiV, I've just been trying to catch up with many past episodes. You're in good company. In TWiV number 5, Vince and guest Saul Silverstein discuss herpes viruses. I'm in my early 50s and had chickenpox before I was 10. Would it, in your opinion, be worth me seeking vaccination against varicella zoster to reduce my chances of suffering from shingles in the future? You also spoke about post-herpetic neuralgia as a symptom of zoster. This brought to mind another podcast I listened to, Dr. Ginger Campbell's Brain Science podcast. In BSP episode 69, Ginger was talking to R. Douglas Fields about his book, The Other Brain, about the influence of the glial cells on the activity of neurons, including their link to chronic pain. Is it known if, as well as infecting neurons, the virus infects Schwann cells and causes them to release neurotransmitters that make the neurons hypersensitive? I have heard that vitamin D can have anti-inflammatory effect and that many people are deficient in vitamin D. Would this mean that post-herpetic neuralgia could possibly be relieved by either vitamin D supplements or creams or by phototherapy with UV light to induce vitamin D synthesis directly in the skin? If chronic neuralgia is due to glial cell sensitization, what other treatments may be of benefit? Thanks for my rambling question. Sorry for my rambling questions. Peter, who says he's a photographer from the UK, now living in Turkey. Vincent, apparently you questioned Saul about this again. Mm-hmm. And Saul says there is no question that the vaccine is efficacious for those over 55 who wish to be protected from zoster. It's not 100%, but it clearly works. VZV, that's varicella zoster virus, is found predominantly in neurons, but there is evidence that it also may be in supporting satellite cells as well. So I got my shingles vaccine, and one of the things that really tipped me over the edge on that was post-herpetic neuralgia. My understanding is that is... So that's when you get an episode of shingles and you recover from it, yes, but the symptoms of the nerve-associated pain don't go away. As if there's been some sort of at least long-term or perhaps even permanent damage that leaves you with the pain, which, as I understand it, can be absolutely awful. And so you're really in big trouble. And so preventing the shingles will prevent that, yes, and the vaccine is effective. I wouldn't... he's got an interesting train of thought here that goes from neurons to Schwann cells to vitamin D to phototherapy and UV light. I wouldn't necessarily be treading down that path. Interesting train of thought, but I just get the vaccine and go think about something else.
Think about the apertures.
Well, and in the middle of the winter, you probably ought to go someplace warm and get some phototherapy.
That's
right. There you go.
That's exactly
try and tax deduct it as a health expense.
Yeah. Got it right.
I'm all for sitting in the sun.
Dickson.
Okay. Attila writes.
Attila.
Or Attila.
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 I met Attila in Brazil, by the way.
You did.
And he said, you pronounced my name right, but I said, which way was right?
We had this discussion last time.
Yeah, we did it in ASV, Rich, remember?
Right.
So it's Attila.
And then we forgot what the conclusion was on another episode.
Not going to do that.
It's Attila, right?
Stop it.
Attila.
Okay.
We've said, I wasn't in the conversation with Attila, but you've subsequently said Attila.
All right, thank you.
So Attila, please write to us if we've got it wrong now.
Well, Attila has already written, and I'm about to read that.
Yes.
Dear Twivers, I have heard many times the myxomavirus and the rabbits in Australia cited as an example of a parasite evolving and causing less harm to its host, and got very interested in the question of who was adapted to what, especially after TWiM-19, when I heard that the Yersinia pestis from the 14th century had been sequenced and harbors very few genetic differences in relation to the strains that circulate today, so that the pathogenicity of the bubonic plague should be in the 14th century Europeans' health, not in the bacteria. As I understand, myxomaviruses from South American rabbits were introduced into Australia to kill the plague of European rabbits that never had contact with this virus. At first, the virus killed something like 99% of the rabbits, but in the later years, it caused less and less deaths, and nowadays it does not kill more. does not kill anymore, I think you meant to say. And this is always cited as an example of a parasite that evolved to preserve the host. But who has changed? Is it a change in virus pathogenicity, host resistance, or both? This made me think that if someone had done the following experiments, if the virus was selected to be less pathogenic, the recent myxomavirus from Australia should not kill rabbits in Europe. If the rabbits had been selected to be more resistant, as may have been the case with pest survivors in the past, Australian rabbits grown with no contact with myxomavirus, with no previous immunity, should be resistant to the American original viral lineage. Do you know if someone has tested this? Thank you again for the great podcast, Attila.
Great question.
Should I read the answer?
Mm-hmm.
Sure.
Okay. Grant answers. I hope that's NIH Grant.
No, that's McFadden.
Okay. Your TWiV listener asks a really great question that gets to the very heart of how virus-host coevolution actually works out there in biology land. Remember, the experiment of releasing pathogenic myxomavirus into feral populations of European rabbits in Australia had been cooking for only about 60 years now, which is pretty long from our perspective, but only a blink of the eye in evolutionary terms. But at least we can say that, so far, selective genetic adaptations are occurring in both the viruses currently replicating in the wild, as well as the host rabbits that are now hopping about in the Australian outback. The original myxomavirus strain released into Australia around 1950, which originated from Brazil and is called SLS, Standard Laboratory Strain, has now evolved into a collection of virus isolates that cause reduced mortality in the current wild Australian rabbits, down from an original mortality rate of over 99% to current levels of anywhere between 10 and 60% mortality. Note that the virus continues to transmit efficiently between rabbits via biting insects, especially mosquitoes, and so has established an endemic relationship, called enzootic, to these wild rabbits. These populations continue to co-evolve, and so we can only report on a snapshot of what we see so far. We know that both the rabbits and viruses have changed genetically by mutational evolutionary pressures. If you take the current field rabbits that have not yet been exposed to myxomavirus and challenge them in the lab with the original SLS strain of virus, they are indeed more resistant to the killing effects and thus tend to live longer with less overall mortality. But, interestingly, if you take current virus isolates from the field and put them into the originally fully sensitive European rabbit strains, the death rate varies from much reduced from the original 99% to even more virulent, i.e. that kills rabbits even faster than the original SLS strain. What this shows is that evolutionary co-selection pressure has generated not only more resistant rabbits, but also a family of virus lineages with very varied virulence characteristics, and these are all competing with each other right now in the wild. We actually don't know which virus rabbit pairings will win out in the long run, but at least we can say that the disease severity we now see in the Australian wild is uniformly dropping from an original near-universal mortality to one that is now much less lethal, but still allows for efficient virus dissemination from rabbit to rabbit. Unfortunately, we don't really understand what the genetic changes in either the virus or the rabbits are really doing at the molecular level. Attempts have begun to fully sequence representative wild virus isolate strains to compare them to the original SLS strain. Lots of sequence variations in these viruses will be documented, but establishing which ones are functional for disease and which ones are silent will take quite some time. On the rabbit side, we know even less. Although the full sequence of the original European rabbit, Orytolagus caniculus, has been reported, nobody I know is currently sequencing the feral virus-resistant rabbits that are now living in the Australian outback. With DNA sequencing costs dropping so dramatically, perhaps somebody out there will one day pursue this scientific question in detail. Maybe even one of your TWiV listeners? Anyway, I hope these points help clarify the question. Cheers. Grant.
Nice.
Nice.
Excellent.
But
that does it.
If someone sequences the rabbits, you won't be able to publish them.
That'll get everybody hopping.
Because they might get out.
That's right. Yeah, this is a really cool story. We have talked about this before, I think, on TWiV, right, Rich?
Oh, yeah.
Yeah. We've talked about the story, and I think we may have mentioned before that the evidence was that there was genetic variation in both the virus and the host to approach a new equilibrium. But this is a much more detailed answer. And yes, people are working on it. And in fact, they can sequence the rabbits and they can sequence the virus, but that won't give them the answer. Okay, this is always the problem. They're going to find mutations. Okay, which one does it, and actually which ones do it. It's going to be a very complex problem. It's an interesting problem, but sorting out virulence factors in any of these creatures is a very, very difficult task.
Did I tell you guys that I once had the honor of meeting Dr. Fenner while he was still alive? He was the one I thought who developed this myxomatosis virus to begin with.
Yes, he was the one who introduced it into Australia.
Indeed, a very distinguished and pleasant gentleman to sit and talk with.
Quite a gentleman. Yep.
Yeah, absolutely.
Not like us.
No, of course not.
Alan, you're up next.
Okay, so Thomas writes, "Hello, TWiV crew. Quick question for Dr. Racaniello et al. In TWiM 21, you mentioned roughly 30 minutes in that viruses that have a long infection time have an incentive to inhibit apoptosis, which you brought up because of its connection to lesser-known mitochondrial functions, calcium regulation, etc., as opposed to only respiration. In a recent lecture I attended, a review titled Hallmarks of Cancer, see attached, mentioned evasion of apoptosis to be one vital pathway in the sustained development of cancer cells into metastatic forms. Is there any way to trace the development of oncogene-associated viruses to inhibition of apoptosis, or do these two mechanisms function independently? I haven't been able to study the full details of cancer-correlated viral infections, but I'd imagine tracing phylogenetic heritage is generally much more difficult in viruses due to the smaller genome size and huge mutation rates compared to the host genome. Does horizontal gene transfer occur among viral quasi-species? Shall we take that question first?
Sure.
Actually, no, the second one... Yeah, this one is independent.
Right. For sure, horizontal gene transfer occurs.
Yes,
among viruses. There's a lot of support for that from the meta-sequencing analysis of the ocean, and so we think viruses do. They swap genes. That's how they evolve. The oncogene question is very interesting. There are certainly oncogenes known to play with apoptosis. So the RAS oncogene, which is one of the ones picked up by a retrovirus, it allows cells to survive by inhibiting proteins that would otherwise cause apoptosis. So a cancer cell doesn't want apoptosis to occur. It wants the cell to live forever. So it usually inhibits apoptotic events, and that's what activated RAS does. That is the oncogenic RAS. The DNA tumor viruses, the so-called DNA tumor virus, adenovirus, polyomas, and papillomaviruses, they encode oncoproteins that inactivate the tumor suppressor protein P53. And that P53 has many functions, but one of them is to stop cells from dividing. If something goes wrong and they keep dividing, this P53 stops them. And it also activates apoptosis as another defense against cancer, basically. Mm-hmm. And these oncogenes inactivate P53. And they say, nope, you guys are not going to do apoptosis. You're going to keep living.
Yeah, so cancer and many viral infections have a similar agenda.
I think that's the answer to that one, right, Rich?
Yep,
Sound
absolutely.
Good?
I think of it as a sort of a tug of war where the virus tries to kick the cell into replication, and the cell says, no, right. I don't want to do that, and tries to kill itself. And that's where the anti-apoptosis thing comes in. The virus has to keep the cell from killing itself. Okay, so it's a sort of a backup to the cell's response to trying to commit suicide.
Yeah, we're going to do this in great detail when we get to our Virology 101 about transformation and oncogenesis. This is just a little hint. It's going to be a lot of fun. I like that one. But that's a ways off. We're at... next one is splicing.
Right.
Okay, go ahead, Alan.
All right, he continues. Question 2. Lately, I've been trying to find an effective algorithm to determine the usefulness of various life sciences textbooks online, or at least a quick way to sort out hand-waving quacks and legitimate groundbreakers. The problem with this is that the review process is often based on a pitifully small sample size, n is often less than 10, and contains concerns over A, the truthfulness, thoroughness of the review, and B, the ability of the reviewer to accurately evaluate what the book has to offer. Given that the average cost of a textbook at or above the undergraduate level is nothing to shrug off, it's a bit of a problem. One such book is Evolutionary Dynamics by Martin Nowak. From what I can discern, the book contains a treatment of theoretical biology from a highly mathematical, by a standard biological perspective, approach. But in being a relatively newer and interdisciplinary set of topics, it presumably has the advantage of being both ignored by spare time-deprived academics and potentially very useful. While I realize the vast portion of his thesis may be invalid once the mathematical obfuscation is removed, default treatment of unknown author is skepticism, his work could easily be legitimate. I write to you about this primarily due to a supposed prediction made about the field of HIV research by Nowak. If Google Scholar and citation number are worth anything, I believe the relevant publications can be found here. Any links to a Google Scholar search on Nowak. If you could shed some light on this topic, it would be greatly appreciated. Thank you for your time. As usual, the shows are fantastic. Keep up the excellent work. And I don't know what the thesis is here that's in question.
Right.
I tried to look up these papers, and he's published, Nowak, he has published a lot on mathematical models of HIV replication, but these are quite old papers from the 90s. And I think it was before we really understood the high turnover of HIV in infected individuals, the fact that you make millions of virions per minute and many, many millions turnover each day. And I think he was trying to model that.
Yeah.
Whether his models were correct or not, I don't know. I wasn't actually able to get to any of these papers. So I can't really say anything about this book.
Right.
It is a problem finding good reviews. I looked at, while you were talking, I just looked at my textbook, and there are just a handful of reviews.
Yeah.
And they're often conflicting, and they don't tell you anything. So,
and as you point out, a book like this, it's a tiny little niche. You've just written to three virologists, and we're looking at these papers that are virology papers, but it's all math, and it's mathematical modeling of virology, and, well, I'm not qualified to evaluate this.
There are certainly people, Jonathan Eisen, for example, would be well qualified to look at this, and others as well.
But that's not going to, unfortunately, those people are busy and don't tend to write reviews of textbooks.
If anyone has read the book, however, we can tap into our audience and write in and tell us if you can answer Thomas's question. All right, that brings us to me.
Right.
Yep.
Tim writes, Dr. R. and esteemed cohorts, I'm hoping that you would take the time to watch this video of Dr. Eric Schadt. You may know the basics and may want to fast forward. This is the current location. Please comment on a couple of items. One, the extraordinary gadget he uses to analyze real time and what it is doing and what he is doing at the molecular level and then at the systems level via computer modeling simulation. And near the end of the video, there is an interesting virology microbiology slide that he shows. He says is based on unpublished data that he should publish pretty soon, so you may want to comment on it after it is published. Thank you for all the time that you put into this podcast. Science education is so important. So this is a video of Eric Schadt. So a little background. He's chair of genetics at Mount Sinai here in New York, but he came from the company PAC Biosciences, which develops, among other things, high-throughput real-time DNA sequencing machines. And so that's what he's doing in this video. He's talking about this very interesting technique where you attach DNAs to a well, and then you can add polymerase, and it will copy the DNAs, and you can read off the sequence by using fluorescent marked nucleotides.
Wasn't this a pick of the week at some point?
Yes.
Because it's an awesome technology.
Yeah, I think the pick was a movie of the thing in motion, not Schadt himself, but the actual sequencing,
Right.
So that's what he's talking about. He never really explains. This was a video from an MECFS conference at Mount Sinai, and I think there was a lay audience there. So that's what it is. It's basically sequencing. And then his whole shtick, if you will, is that you can't just look at the sequence, but you have to combine it with other information to really learn about diseases, like what RNAs are expressed in different tissues, the protein-protein interactions. So he gets all these data from diseased people, for example, people with diabetes or other conditions, and then you sequence in each organ the DNA, you find out what RNAs are expressed, you find out the protein-protein interactions, and then he tries to display it in three-dimensional charts, basically. And then he says you can pick up trends which then point you in therapeutic directions. Is that right, guys? Do I got it right?
You got me, because I didn't actually look at the video. Right. Okay.
So I did.
I think that's the idea. Yeah.
I did, and that is it. And then towards the end, he actually talks about sequencing not only you and your microbiome, but all the microbes around you. And he has this vision of sequencing neighborhoods and having all the data online, like what microbes are in this house versus that house and the other house.
The envirobiome.
And there was a very nice New York Times article about this a few months ago, where they try and do this on a very local level. I mean, he showed a slide where, a kid says to his mom, "Can I go to Johnny's house?" So she calls up on her screen the sequence of all the things in Johnny's house, and she goes, wrong bacteria, blah, blah, blah. You can go to his house, but don't stay for dinner,
That's right.
So
don't flush.
He's thinking that someday we're going to have all this information.
And it's, as I said, it's an awesome technique and an incredibly clever technology. But the one thing to bear in mind with all these types of super high throughput sequence the world experiments is that the signal to noise ratio in these is ridiculously low.
Wow.
The signal, you're going to find bazillions of things everywhere.
Right.
And you put all these into a database, and the database is mind-bogglingly huge. And then you're going to try and pull out correlations. It's the statistics behind this are really, really dicey.
Yeah. One thing I found interesting. So, Tim, I don't know what you mean about his discovery that he's going to patent or publish. I don't know what that was. I couldn't pick that up. But I did notice one slide. When he was at Pac Biosystems, they sequenced surfaces throughout the company just to see what was on all the surfaces. And he showed a slide of all the flu strains that they picked up. And he said you could tell there was a flu outbreak because they started isolating all these different strains. And on the slide were some H5N1 strains.
They should never be allowed to publish that.
He never mentioned it. He didn't say anything about it. It's just there. So I thought that was interesting. Does that mean that they are circulating maybe and we don't know about it? Or is that a mistake? I don't know. Anyway,
it means there was a chicken in the lab.
I thought that was cool. Yes.
That's right.
Rich, you're next.
Lance writes, "Dear doctors, I just finished TWiV 160 with Dr. Moore." That's where we talked about cancer viruses. Great interview. I knew Dr. Moore and his wife, Dr. Chang, while I was doing my MS at University of Pittsburgh. Great people, but I'm really disappointed I never knew that Dr. Moore had an EIS background. That's the Epidemiologic Intelligence Service with the CDC. I have had an interest in the EIS for many years now and hope to apply soon when I finish my PhD, but who really knows how and when that happens? I know you have touched on the EIS program a few times, but it would be great if you could bring on some of the virologists who have made it famous. For example, Joseph McCormick, C.J. Peters, Peter Yarling, etc. A couple of messages to relay. First to Dr. Despommier. The Vertical Farm Project is alive and well in Chicago. I'm sure he would be proud. And second to Dr. Condit. Good luck with his personal genome project spit collection. I sent mine in about a month ago. Very interesting. Thanks again for all the great work you're doing. Peter. Lance. So, yeah, we ought to get some more EIS people on. It's a great program.
Yeah, it is.
And we ought to do that. We were going to do the book Inside the Outbreaks. Get the author.
One of these days.
Do that.
Yep.
And so I got a little footnote to the Personal Genome Project. I think that I said that both myself and my wife had sent our spit tubes in, but also our identical twin daughters have done the same thing.
Roger.
That's good. That's good. That's why you got in, because of the twins. Yeah. It's cool. Great.
Cool.
Dickson Despommier, you're next.
Peter writes, Dear TWiVsters, have you seen this picture of a T4 phage tattoo? And he gives a web address. In 2007, science writer Carl Zimmer, having seen someone with a DNA tattoo, wondered how common science-inspired tattoos were. He asked readers of his blog, and their response was massive. Getting back to the "Are viruses alive?" question, I was having this discussion the other day, and someone came up with one of the best answers I have ever heard to the question. Quotes, "It's a bit like asking whether a photon is a wave or a particle." Very thought-provoking.
Okay.
A photon is both, right?
You're going to say both.
Yes.
Exactly right. The answer.
So a virus is living or not depends, maybe.
Yeah.
When it's in the cell,
Yeah.
it's living. When it's a particle, it's not.
Yeah.
It's a zombie, right?
It's a zombie.
This T4 tattoo
It's cool.
is, it's creepy.
Yeah, see this?
It's got almost like crab legs.
It's
something.
Very
pretty.
creepy. It's a huge tattoo also. It's not tiny.
Yeah,
You have to really like T4.
I guess.
Wow.
Oh, I don't know. Nobody's going to mess with you with a tattoo like that.
So the person who has it says he or she works on, it's a postdoc working on prions, but the prion structure isn't known, so he or she put a T4 structure on his or her arm. Cool. Thank you, Peter. That brings us to Dickson.
No, I just did one. I'll do another one if you want.
No. It's okay. Alan is next. Sorry.
Yes.
So wait,
What?
Oh yeah, I see. There's a link down there to Zimmer's book, Science Inc. Tattoos of the Science Obsessed.
Yeah, he now has a sort of coffee table book out that's portraits of these different science tattoos.
Yep.
You can even look at it on Amazon. Some of it. Holy cow!
We just lost Rich.
He's gone. Okay, Alan, go ahead.
Okay, Allison writes, Hi, TWiV gang. Love the podcast. I'm a graduate student at the University of Washington, and I've found your podcast both fun and informative as a student of virology. I was wondering about a technique that was mentioned in episode 158 at the University of Michigan. Rich mentioned mouse blots when speaking with Kathy Spindler. I tried looking up this technique on PubMed and found a technique used by Luis Villarreal called fluorography of whole mouse. That sounds almost like a dish in a French restaurant, doesn't it? I can't seem to find the publication in which Dr. Spindler describes this technique. Could you or Rich help me find it? Thanks so much for this awesome service for the public. And I guess you forwarded this to Kathy Spindler, who replied, The paper in which we used whole mouse blots is Ball et al., 1991, Virology 180:257-265. We can link to this in the show notes. There are some other references in here, and we'll just put links in, I think.
Right.
We use P32 probes, which don't give as high resolution as some other isotopes, for example, tritium used by Villarreal in the paper that Allison cites, but which uses a different approach, in situ autoradiography and whole mouse fluorography. I think some modern isotopes, P33 or S35 labeled probes, might give better resolution than P32 with whole mouse blots. We haven't used those techniques since the early 90s. And I think you're right that Louise did invent the technology. The earliest paper I can find is Dubensky et al. In 1984. Figure 1 shows why we call it slice mice. Not sure you should say that on the podcast, though. Maybe it sounds offensive to people.
It's okay.
But all right. Ian Lipkin, Louise, and Mike Oldstone are co-authors on a methods paper that I would have to walk to the library to get. Horrors, it's 5 minutes away inside the building. But I've ordered it as a PDF online from the library, and that should come in a day or two. The modern world.
Tell you.
I wonder if it will have a historic perspective on the technique. That's a 1989 paper. I can send that to you and or Allison once it comes. I can also send her my bench protocol if she wants. These papers might have more detail than ours, where we merely cite Dubensky et al. And Rochford et al. However, I note that we did mention that we had the microtome sitting in a Sears chest freezer. Kind of a useless detail.
Cool. This is very geeky for most of our audience, I think. But I think it's a good idea of connecting researchers by TWiV.
Sure.
Yeah.
Yep.
I in about 19...
Don't get carried away with it, though, okay, folks?
I think it was about 1976 I visited Luis at his lab in Irvine, and he showed me his mouse slicer. It was a mega microtome that he kept in a freezer. So I wonder if everybody out there can visualize this or even knows what a blot is.
Right. So the idea is you're gonna radio label and then section a whole mouse, right?
Actually, it's the other way around. You take the mouse and embed it, okay? And then you section the whole mouse. So we're talking about huge sections. And you take an individual section.
For those non-lab geeks in the audience, you're putting this thing through essentially a scientific deli slicer.
Right? Yes, exactly. Yeah, very good. Deli slicer is good. And then you take an individual slice and you lay that down onto a piece of nitrocellulose and you allow buffer to seep through the mouse tissue and through the nitrocellulose. And what happens is that the nucleic acids, RNA and DNA from the mouse slice are transferred from the mouse tissues to the nitrocellulose. So you basically have a replica of the mouse slice of both the DNA and the nucleic acid and the protein, for that matter, now fixed to the nitrocellulose. And that allows you to take that nitrocellulose and then soak it in anything you want that will detect whatever you want that's on the nitrocellulose. So, for example, you can soak it with, if the mouse was infected with polyomavirus, which is the experiment that Luis did, you can then soak it in something that has a radioactive piece of polyomavirus DNA in it, and that will hybridize to, that is, bind to any polyoma DNA that's on the nitrocellulose that came from the mouse. And so you have an image of where the polyomavirus DNA was in the mouse on this slice. So it's a quick way of doing virus pathogenesis. You infect the mouse with the virus, do these blots, and you can tell from the blots where the virus went in the mouse.
Well, I don't know if I would call it a quick way, depending on how many slices you make.
Depending on how many slices you make. Well, yeah. Maybe most of the things that I see just look at a couple of sections from that are representative of the tissues. Cool technique.
Yeah, it's really neat.
Isn't something being done like that with fluorescent green protein also?
No, but it is being done with green fluorescent protein.
I meant green fluorescent protein. Come on.
I'm just giving you hell.
In that way, you don't have to kill the mouse. You just fluoresce them and find out where it is.
Oh, you could do that. You could do it real time with a live mouse. Yeah.
There
are lots of imaging ways now that you can do it on live mouse. You don't have to slice them anymore.
Right,
yeah.
You might not get the same resolution.
Yeah, the resolution is not as good.
But Dickson's right. That's the modern equivalent of the same thing. Yep. Is using basically a labeled virus. Yep.
Right.
All right, the next one is from Danielle. Hello, TWiV. First, I would like to say I love the show. You guys make repetitive but still awesome lab work go much faster. Before I get to my silliness, I'd like to ask a serious question or two. If I buy the Microbe World app, you guys get a good cut of the profits, right? No.
We don't see a dime.
We don't. It goes to ASM, the American Society for Microbiology.
Right. Which is a worthy organization.
Indeed.
Yeah. You could support them. However, Danielle, if you would like to listen to other podcasts besides, TWiV, TWIM, TWIP, and the others that ASM puts out on that app, you can go to Stitcher Radio, which is free, and then you can download any podcast in the world on it. So you don't have to pay anything. I shouldn't say that because ASM does help us out, but
that's true.
There are alternatives. Also, were any clustering analysis done on the H1N1 strain that was sequenced? If so, did it prove that the strain jumped from swine to humans in Mexico? Sorry if the answer is common knowledge. I'm only up to 2009 in your podcasts, besides the few recent ones I've listened to. Should I spoil it for her?
Yeah.
Go for it.
Go for it.
Well, it seems very likely that the 2009 pandemic strain did originate in Mexico. Whether it jumped from swine to humans or from humans to swine to humans, I don't think we'll ever know. We don't really know where the reassortment occurred. But I think the origin is most likely Mexico, yeah. Anyway, I'm not sure if you guys are aware of Reddit, but they just discovered that the Sputnik virus can infect mammavirus. So in a Redditor fashion, I made this meme and thought I'd share it. So she puts a link to the Reddit thread. And Reddit is a site where you can basically post very long discussions, right? I don't really go there, but that's what my son tells me.
This is news to me.
They have a comment thread on Sputnik and mammavirus, which is very interesting because everyone's astounded that this happened, and there are a few scientists in there. So that's Reddit. And then she says she made a virology-related meme. Now, everyone knows what a meme is, right?
Keep talking.
You don't know what a meme is?
This is a word that I've encountered only recently, and I have a hard time defining it. We'd better do that.
Well, it was coined by Richard Dawkins, wasn't it?
Oh, really? That I didn't know.
Really?
I think it was. He came up with this idea of a meme is an idea that spreads in an almost infectious fashion among people.
You're right.
So
he did
some
coin it.
Joke or idea that just everybody hears
Got it.
in a very short period of time. It spreads virally.
Yeah, he coined it in 1976 in The Selfish Gene.
There you go.
Okay.
So, in fact, when people talk about viral videos or something
memes,
that went viral on the internet, it's all based on that concept of a meme.
So, Danielle made a meme, a virology-related meme, and we'll send you the link to it so you can look at it.
It's a reference to another meme that I have seen elsewhere, but I don't know what you're
Yo, dog, I heard you like viruses, so I put a Sputnik virus in your mama virus so you can infect while you infect. That's cool. Thank you, Danielle. You didn't waste our time. It's
Not
okay.
at all. No, we got plenty of room for silliness.
She said, I hope I didn't waste too much of your time. On the silliness, no, we appreciate everything.
You wasted just the right amount of time.
Exactly.
Thanks very much. All right, Rich. Oh, this is perfect.
Hi, Vincent.
Who is this?
Oh, Richard writes.
Richard, yes. That's right.
Hi, Vincent, Dick, and other hosts.
At
all.
At all.
I have a question that may be a little outside TWiV's remit, but at least partially applies. Recently, I have heard in podcasts via Stitcher Radio about the possibility of genetic engineering of humans and restructuring the woolly mammoth.
No, resurrecting.
I'm sorry, resurrecting the woolly mammoth.
Please.
Restructuring, resurrecting. Some of the methods mentioned include cloning and things outside of virology. However, I was wondering if viruses, particularly retrovirus, would allow genes to be edited, removed, or inserted. If so, would this be practical, or do better methods exist? Would it even be possible to engineer a retrovirus so as to insert a gene or other DNA reliably into a host? Would this be a temporary change or inherited by any offspring? My guess is that inserted genes would be inherited as long as they were present in germ cells, much as the remains of retrovirus infections remain in many genomes. Also, could a retrovirus be used to edit the genome of an elephant embryo so as to make it match, say, a woolly mammoth? My guess is that retroviruses aren't that big and tend to insert things randomly. I'm thinking that would be a showstopper.
I'll say it.
That's great.
Some random speculation. I wonder if it would be possible to engineer something like a ribosome that would edit DNA given a template, either DNA or RNA. So it would run along a DNA and compare the sequence to the template. On finding a match, it would then edit the bases and match the rest of the template. Thanks for the podcast. I listen to every episode and like the way you simplify things enough for a non-virologist such as myself. I'm a water and wastewater engineer. Schmutzdecker.
There you go. There you go.
Looking forward to more virology. Regards, Richard.
We have an awesome audience.
We do.
Yes,
we really do.
Absolutely.
I think it is great that we've got somebody who describes himself as a non-virologist who has just outlined gene therapy, germline modification, and site-directed mutagenesis without
simply by listening to
formally
TWiV.
been taught any of them.
Oh no, he has a formal education. He's been a TWiVer.
Right. Yes.
So, Richard, we're quite a ways from recreating a woolly mammoth or engineering an elephant, but in principle, everything that you suggest is possible.
Yep.
Okay. Let's see. Retroviruses,
and is in fact being very actively worked on right now.
Retroviruses can be used to insert genes into genomes, and the change can be permanent if it goes into germ cells, and that's being used to treat diseases such as immunodeficiencies. And actually, with respect to editing, what comes to mind is the zinc finger nuclease, that you can use to very site-specifically engineer DNA. So it's all right on.
Yep.
So he's right that the retroviruses are too small to carry large pieces of DNA. But for jingle genes, it's okay.
Yep.
What I wanted to tell Richard is very cool now. So what you want to do in gene therapy now is you want to take stem cells out of the patient and put the gene into them, say, using a retrovirus to correct a deficiency and then put them back.
Right.
But what you can do now with almost any type of cell is to turn them into a stem cell. In amazing work that was done by a Japanese fellow a number of years ago, he showed that with just 3 plasmids, you can take any cell, a skin cell, whatever, and put those 3 plasmids in, and they become stem cells.
Well, they become pluripotent stem cells. There's still some debate about whether you can really differentiate them fully into everything, but it's a hugely important area of research right now.
So that's a way to put genes in.
Yes.
This idea of using a ribosome to correct, I think you can use polymerases to do that, which already have editing functions, right?
Yeah. Right.
So the other thing is, if you wanted to make an elephant into a woolly mammoth, you have to take the whole mammoth genome, and
you need a really big wig,
and then take out the nucleus from the elephant cell and replace it with that of the woolly mammoth. Because if you have the elephant DNA in there, it's going to confuse things.
You just have to titrate the dose of Rogaine.
That's right. I don't think on this show, we did Venter's experiment where he took a mycoplasma genome, about a megabase, and put it together and then stuck it into a cell and got mycoplasmas out. So he assembled it in yeast and then put it into a mycoplasma and got it out. So this can be done at that scale, but bigger is tough.
Yeah,
it'll happen though. Everything can happen. Right?
My turn.
Right, Dickson, Despommier.
Richard,
I think this is from the same person.
Oh, okay.
I think so. Yeah.
You do, eh?
Yeah.
Richard writes, "Hi, Vincent, Dick, and co-hosts."
Yeah, it is the same guy.
Same guy.
Sorry about that, guys. I've just listened to the Science Show podcast from the 3rd of December. They mention exosomes and membraned container that is secreted by cells. These contain genetic material, RNA, as well as a number of other proteins. I had no idea that the human body contains such a horizontal transport mechanism. I can't help but compare these to a virus. Though there are clear differences, do you think they are related? Maybe cells learned this trick from viruses, or the other way around. Thanks again for your podcasts. Regards, Richard.
Can we get this guy into a lab already?
I think he is, this wastewater engineer, man.
Yeah, I mean a virology lab. Richard, you get it.
That's right.
Yeah.
Must like TWiV.
Yeah.
So these exosomes are really hot. They are pretty recently discovered, and they have RNA in them, and we're wondering, are they transporting things among cells? But they're produced by the same mechanisms that viruses bud from the cell surface. Mm-hmm. Little containers of membrane-bound containers bud off by the same cellular mechanism. Now, who was there first? Well, the viruses took advantage of the cellular machinery to bud out of the cell, so that's what's happening here. It's probably the same mechanism or similar as the exosomes.
Right.
My guess is the virus ripped it off from the cell.
Yeah, probably.
Yeah. Yeah, because you see it in so many different viruses that are very evolutionarily distant. It's got to just be a mechanism that's sitting there waiting to be exploited.
Alan, you are next.
Jimmy writes, "Hey guys, I discovered your podcasts a few weeks ago and have really been enjoying them. I toyed with biology as an undergraduate and I love it, but I ended up in business. I just learned of the Science Exchange today and thought you might be interested, given your interests in using the internet to advance science." And he provides a link. We probably should have made this a listener pick, right?
Yeah.
Yeah, I already have 3, so I'll put it down in the listener picks. Sure. This is a cool site. If you want to do an experiment, you put it here and someone will do it for you.
Yeah.
They have 1,000 scientific service providers.
Right. Now, these are contractors who who do the sort of outsourcing work that is now very common in biological research. So if you need somebody to do your DNA sequencing, you don't need to do it yourself. They've got people who will do it for pennies a base.
So
you just search for DNA sequencing.
I'm putting in clone H5N1. Let's see what we get. I'm on an H5N1. We couldn't find an experiment with that name.
I put in mutagenesis and got site-directed mutagenesis. But they said they didn't find anybody. I can't believe that. They also say they want me to sign up.
Yeah, this is...
Maybe I got to sign up.
You got to sign up before you get anything.
Right.
All right. It's a good idea, though.
Yep. Actually, hiring somebody to make your clones nowadays...
Yeah, that's the way to go.
...is really worth it.
Yeah. Yeah, there are plenty of companies that do that.
Yeah.
You shouldn't waste your time cloning anymore.
Right.
Alan Dove is gone. Dickson is gone. It's you and me, Rich.
Dickson is gone.
Yeah, I think he went nature called.
All right. So you want me to keep going here?
Who read this last one? Jimmy? Alan?
I think that was Alan, yeah.
Yeah, so I'm next. Juanjo writes, Dear Vincent, Dick, Alan, and Rich, while waiting for the launching of a variant/sequel of TWiV to be broadcast in Spanish, as proposed by Estanis Nistal, with the enthusiastic support of several other Spanish-speaking listeners of TWiV, I have submitted a short notice for the journal Virologia, the Bulletin of the Spanish Society for Virology, SEV, Sociedad Española de Virología, to divulgate your netcast. This note was published in Volume 14, Number 3, and he provides a link. This note intends to inform the virology community in Spain of the existence of TWiV, including links to virology blog and TWiV web pages. I hope this information will contribute to spread the word about TWiV among Spanish virologists, students, and the general public. Thank you very much.
Great.
Hey there.
I'm back.
And you're back, but Dickson is gone.
Oh, are we mutually exclusive?
No, he had a Skype problem. It's a joke.
Yes, I figured.
It's horrible when you say a joke and no one laughs, although Rich did chuckle.
Yes. Yeah, it's part of my job.
Rich, you are next.
Peter writes, Dear doctors, I'm sure others will send you these links. And there's 2 links here about norovirus infections on airplanes. About a series of norovirus infections on an Air New Zealand plane. There was inadequate cleaning and disinfection between these flights. So somebody got sick on a flight, and in subsequent flights, a lot of other people got sick. So it's the cruise ship thing in the sky.
They vomited. That's the problem.
Yeah.
They threw up probably in one of those bags, right?
Yeah.
And that aerosolizes the virus, as Stephanie told us.
Yeah.
I have several times come down with fairly severe chest infections, one requiring hospital treatment. After flights, it is, of course, difficult to know exactly where I acquired the infection, but at the airport or on the aircraft seem the most likely places to be exposed. I'm a regular listener to TWiV, TWIP, and TWIM. Keep up the good work. These are all fascinating and informative shows. So, it seems to me an airplane is a wonderful place to cram people together and circulate all of their organisms from one to another through the air. You've got a comment.
It's kind of a setup. And in fact, the air exchange system there, it's the pressurization inside the cabin requires a certain amount of fuel to operate. So it's in the airline's interest to not have the air circulating maybe as fast as possible. But even if they could, you've got everybody crammed into this little space together. And there's also been some research on the water in airliners' water tanks that a few years ago, somebody just decided, hey, let's take water samples on airliners and see what's in it. And they found coliform levels that were outside FDA and EPA standards. And what subsequently transpired was that they discovered that there was this sort of regulatory logjam. The FAA thought it was somebody else's problem. The FDA thought it was the FAA's problem. The EPA thought everybody thought everybody else was taking care of it. But I gather the EPA in the United States has issued rules now for disinfecting these water tanks. I don't know if those have been applied to airlines in other countries. I will say that after seeing the original research, I stopped drinking the water on airplanes.
There have been many documented in the literature outbreaks of, say, influenza on airplanes. Very high attack rates, over 75%. It's a very efficient way to get infected.
Yep.
And actually that figures in quite a few epidemiological stories that the people shared a flight.
You bet.
There's not much you can do if you want to travel. You have to take the risk.
Even cruise ships have their
but I don't need to take. I don't need to take a cruise ship. That's for sure. But I do need to take airplanes.
Right.
All right, Dickson, you are next.
I just stepped back in the room, so you're going to have to inform me where we are here.
Well, you'll have to figure it out, Dickson.
Oh, thanks.
ESSA, E-S-S-A.
Oh, ESSA, of course. Dear TWiV team, happy holiday and happy New Year.
Thank you.
Thanks a lot for the great virology blog. I would like to know if there's any possibility to any virus to be fused with another one to be infected by another virus. As we know, the infection concept is just a conscious term or scientific term to us since it causes us to be sick, unwanted health condition, due to our reaction to microorganisms such as viruses. If viruses are independent, not related to us organisms, how would they get their compatible receptors to ours on our cells? In Nasia mine, a mine is best known for its extraordinary selenite crystals in the Mexican state of Chihuahua, virologists have discovered millions of viruses in many samples from underground water and crystals. The temperatures in such a mine is nearly 50 degrees and the humidity is nearly 100%. After sucking out the emerging water in the cave, roughly for a million years ago. Maybe these conditions are suitable for the basic molecules for any living cells, such as amino acids, which are the basic building blocks of viruses. What if more than aboriginal or primitive viruses, or I'm sitting here without my glasses because I stepped out of the room and left them outside, so please bear with me here, or primitive viruses, separated molecules fused to form a single genetic material to generate new generations or advanced viruses, consequently forming more advanced cells to form the aboriginal creatures such as bacteria and abiogenesis.
Can you go get your glasses, please?
It's too late. I've just read the letter.
We might have another one.
What she's probably trying to ask is, can life arise from non-life given the right conditions? And given billions of years, the answer is yes. Given millions of years, probably not.
Right.
Can a virus infect another one?
Well, we just had an example of that.
Yes.
We had the Sputnik.
Mimi Mama.
Sputnik. That's right.
Well,
I still hesitate to call that a virus infecting another virus.
No, it's a piggybacker. It's a piggybacker.
It's a satellite virus.
Yeah. That's right.
So it cannot infect the cell unless the other virus is infected the same cell.
But
it's not really infecting the other virus.
Yeah, it doesn't infect the particle, but the virus infects itself. For sure, that's the Sputnik virus, ESSA. Right. Which we have, you could find that in the blog somewhere.
Yeah, I think, in principle, what she's suggesting is the argument that was generated back in the days of Louis Pasteur, who tried to disprove spontaneous generation, but obviously had to account for life on Earth in some way. Yeah. So his flask, by the way, is still sitting in the Pasteur Institute, and it's still sterile. And it's still loaded with nutrient broth, waiting for a microbe to crawl up that very long glass tube and infect it.
Yeah. I think it's unlikely that life began as viruses, given that viruses need something to infect.
Exact.
But I think, along with the first life, very early on, viruses appeared as a byproduct.
Hey, we only have 2 more left. Let's do it, Alan.
Okay, so Joe writes, all my family members work in hospitals. There are many excellent MD podcasts already. The thing I like about TWiM, TWiV, and TWIP is precisely that they are different from those. Having a panelist who is a doc working in emerging diseases is interesting. Local doc stuff is not what I listen for. I listen to every episode of all 3 shows, by the way. Thanks for what you do.
Oh, cool.
Cool, thank you.
I'm glad you like it. All right, our last one is from Tom. Dear Twivians. That's a new one.
Yeah.
I've been listening to TWiV podcasts at lunch for over a year. My anthropology degree from 40 years ago did not prepare me for TWiV, nor did my 30-plus year computer software career. However, I've been reading Scientific American since the early 60s, and I subscribe to Science News, so I'm not completely lost.
Right.
All that being said, I heartily commend you for your excellent presentation of this subject matter in a way that helps us layfolk follow the story while stimulating enthusiastic participation by your visiting specialists. Bravo. Thank you. Oddly enough, my grounding in anthropology is what prompts this comment. I hear lots of interviews on all kinds of subjects on radio, TV, and podcasts. I can tell when a researcher is being interviewed because almost every time they begin to answer a question, they start with the word so. He's so right. Sometimes it's part of an opening phrase, but more often it appears to be a sentence all by itself. And you Twivians tend to follow the pattern when beginning your follow-up questions. Replay a segment of one of your podcasts and count how many times you hear paragraphs start with so.
I don't have to. I've noticed this myself.
Yes, me too.
Everybody does it.
So what's wrong?
Since anthropologists are not forbidden to ask why questions, I have a hypothesis as to why this happens. A researcher's frame of mind is one of gathering evidence for chains of causality. This and these get together, so this happens. This pattern of thought may be carrying over into their patterns of speech. Researchers think a certain way, so they speak a certain way. So keep up the good work and try not to be too self-conscious about starting your comments with so. I think it's just a filler. We don't like emptiness, so we fill it in before we get going.
And I think it usually is a preface to, I need to tell you this story so I can tell you that one.
That too, yes.
It's usually because there are so many nuances and complexities to it that we'll pause for a moment and then say, so thus and such happens.
Mm-hmm.
Yeah.
And then he writes, I found TWiV from a link on an MECFS website when I was researching my wife's illness. The variability of the symptoms, not just between patients, but from day to day for one patient, has inspired my wife to refer to her illness as WBD, weird butt disease. She can never predict how she will feel in a day or even in an hour. Occasionally, when asked by an acquaintance what illness she has, she says she's come down with WTF. Yeah. Recently, Vincent theorized that the illness may have some kind of environmental trigger that in vulnerable individuals initiates a cascade of changes over a period of time that finally presents as something we characterize as CFIDs or MECFS or any of the other spectrum illnesses. At that point, there's no hope of identifying the long-gone trigger. Although I find this theory very disheartening, my wife's medical history would not rule it out. She dates the major onset of her illness to the spring of 2005, but looking back, she sees that many of her symptoms began to appear up to 15 years earlier. Vincent, I fear that you may be right. Tom is from Texas. I don't know. We'll see.
WBD. You know, they have there are issues about the names of this disease. Maybe it ought to be, that would address all of them. Call it weird butt disease. I like the fact that his wife has a sense of humor about
You're going to get letters, Rich.
Oh, no.
I'm just kidding.
It wasn't my idea.
That was a very nice letter, Tom. Thank you. I'm glad that you stuck with TWiV after finding it, searching for MECFS info. So I've always wondered if people who arrived here through MECFS, XMRV mainly, have left or if they are staying. Mm-hmm. But here we have at least one that's still here, and I think there are others, so that's great. Mm-hmm. All right, science picks of the week.
There we go.
Dickson Despommier.
I have one, and I can preface this by saying so. No, actually, I won't do that. I will pick a movie that my wife and I actually enjoyed watching from start to finish while laying in bed on New Year's Eve.
Too much information.
Do we want to hear about this?
No, you do, because it was a joyful movie. It was called Creation. And it was a British production of the middle life of Charles Darwin. And it involved some wonderful acting. And if I could tell you who the actors and actresses were, I would, but I can't remember
Paul
their names.
Bettany and Jennifer Connelly?
Yeah, probably right.
Oh.
Did you see the movie?
I've seen this.
It's a fantastic movie. The guy even looks like Darwin. No, I'm not kidding. He does. He has a striking resemblance to the middle-aged Charles Darwin. And, of course, he goes through all these machinations about whether he's going to publish or not, and his wife is a strict Presbyterian.
Yes, I have seen this.
And it was really beautifully done. And my wife and I were charmed by the whole movie. So I picked that Creation.
Thank you, Dickson.
They, as I recall, they portray him as a tortured individual.
He
right?
was, and he certainly was.
Rich Condit, what do you have?
I just got this afternoon from Nicene, and it is a YouTube video of Neil deGrasse Tyson,
yes,
talking about sort of the future of science globally, or our investment in science globally. And he's got a map of the Earth that he starts off with is just a regular map of the Earth with different regions colored different ways. And then he talks about the investment in science in different parts of the Earth, and has the map morph into the countries taking on a size that represents their investment in science. And he does it two ways. One, peer-reviewed publications in the past little while, okay, and or overall, and obviously, well, not obviously, but the U.S. shows up as having a huge investment in science, as does Japan and Europe, with almost nothing in Africa and very little or much less in South America. But then he talks about the rate of growth of the investment in science globally, and evaluated that way, what you see is that the U.S. Rate of growth of science is much, much less. Okay, whereas European, Chinese, and Japanese investment in science is growing. So it makes, in a very graphic way, the point that we've been making over time, which is that the investment in science in America is very much on the decline. So there you go.
Very good. Alan?
Well, I actually had a different pick, but then I saw Rich's pick, which is such a downer. I figured I would pick something humorous this week. It's Out of Context Science, and this is a silly little blog that is often quite amusing, where this person just posts quotes from scientific publications or articles about scientific publications, completely out of context. So the first post is, quote, "Only when the animal pops out a glowing fecal pellet do the bacteria become visible again."
It's great.
Just, no context, just right there. So it's good science, badly quoted is the tagline for it, and it's fun.
It's so great. All the things that you can find on the internet.
Yes.
Neat. My pick is something that Steve Goff passed to me the other day. It's called The Top 10 Innovations of 2011 from The Scientist. Very cool stuff here, like a pocket microscope.
I need one of those.
Well, it uses a cell phone camera. This is a huge thing to do now, is try and use cell phone cameras, which are ubiquitous.
A mouse imager, you put the mouse in it and you get a whole body image, which is $295,000. PCR in a pouch, portable PCR, single cell mass cytometry, a lot of other cool things that you'd love to have, but are awfully expensive. But just show you how neat the technology of science has gotten. There's just a lot of stuff, and there's more stuff that's not even here, which is amazing. So that's pretty cool. Check that out.
Yep.
All right, we have 4 listener picks of the week. The one we just mentioned from Jimmy, that's Science Exchange. And then we have one from Jim. Heard a TED Talk, which I enjoyed a lot. It gives a new way of looking at information to see if life might be present. This has shifted my vote to thinking viruses are alive. It's a TED Talk. 73 from Jim.
Yep.
Cool. Thank you, Jim.
I listened to the TED Talk. It's pretty interesting. It's good.
Next one is from Tim, who, my pick of the week, Patient Zero from Radiolab. It is pretty darn good storytelling, he says. It is a episode of Radiolab where they go through all the different patient zeros that have been studied over the years. Typhoid Mary, the individual who started the super spreader of HIV, etc. So that's pretty cool. And then Mary. Hi all. Over the past TWiV episodes, there has been increasing talk about what happens on a day-to-day basis in the life of a scientist, grad student, postdoc, PI, etc., and what life in the lab is like. A book I would recommend to those curious about this is Natural Obsessions by Natalie Angier. I was given this book by my boss when I was working as a technician in a lab after college and trying to decide if grad school was in the cards for me. This book is based on the true story about the labs that were instrumental in isolating the first oncogenes in the early 80s. It's a great science story for those who have never heard it, and also gives a very good idea of what life in the lab is like at all levels. It also emphasizes what was said recently in TWiV 161, that life in the lab is very much about social interpersonal interactions for better and sometimes for worse. I don't know if this book has ever been picked before, but if not, I would highly recommend to all as a good read. I think it might have been, but it
I think
never...
we picked it before, but it's worth picking
Never
again.
hurts. It's a great book. We've picked... Many things have been picked twice. It's okay.
Yep.
As we say in teaching, a repetition never hurts. Right?
There was actually one of those New York Times videos on their website recently that was a short interview with Eric Lander, where he emphasized what a social activity science is. And I don't think that can be emphasized enough. It is very much a social activity, which comes as a surprise to a lot of people. But it's a good point about this book.
Yep, sure. It is. And that's what we do here at TWiV. We social.
Bingo.
Absolutely.
It's not a verb, right?
You know,
it is now.
If we had
please don't verb now.
Dickson, what are you going to say?
What I was going to say was that if we had all been research scientists in adjacent laboratories to one another, I bet you that we would have all published co-authored papers together, because we do get along well, and we do share information, and we're not egotists with regards to who owns what. You shouldn't feel that way, because you're trying to discover the truth. Nobody owns the truth. So, I mean, I'm amazed at how collegial we are with regards to not just the way we talk about information from some other people's work, but also about our own. We're certainly willing to throw it up for grabs and ask, how much validity is there in all of this? And I think that's a remarkable characteristic. You don't find that too often. And if you look back over your years in science and then put all the names of the people that you've collaborated with, you can see those threads of sociology weaving in and out. And it's quite a wonderful thing to look back on, and I guess I can do that now because I'm retired. But it's amazing how many different people you get to know and get to meet and interact with. And they're all different, and they're all interesting, and it's what makes your own life worthwhile.
You're going to shed a tear, Dickson?
No, I'm not getting maudlin over this.
I'm just kidding.
I'm just getting reminiscent.
One of the characters in Natural Obsessions is Robert Weinberg, right? And he used to go to the lab meetings that I went to because he shared them with David Baltimore. And I remember at my last lab meeting before I came here to Columbia, I was presenting this stuff, and I said, that's my last lab meeting. And he said, would you like me to shed a tear? Never forget that. He's so sarcastic. Jeez. That has been TWiV number 165. You can find us at iTunes, the Zune Marketplace, and TWiV.tv. If you do subscribe in iTunes and you haven't done so before, please leave a comment. It helps to keep us prominent on the iTunes directory.
And we do like to be prominent.
We do. This past week, TWiV 164 was number one downloaded podcast for quite a while on iTunes. Now that's
in our category.
Yes, in our category, which is 2,756 level down. No, just kidding. That's fine. But it's transient, but it's good when it happens. Because if someone sees, oh, look at this top podcast, then they go to subscribe and we get more and more. And it becomes a meme.
Yes.
Right.
Is a virus a meme? Dickson laughs.
I'm just laughing. I'm just, beyond philosophy.
You can send us your questions and comments to TWiV at TWiV.tv. We love to get them. We went through all of them today. It's great. Good job.
Awesome.
Dickson Despommier is at verticalfarm.com, medicalecology.com, and tricanella.org. Thank you for joining us, Dickson.
Pleasure.
Always good to see you.
Yep.
Alan Dove can be found at alandove.com. Thank you, Alan.
Always a pleasure.
Good to have you. And Rich Condit is at the University of Florida in a small town called Gainesville. Thank you, Rich.
Go.
Thank you.
You're quite welcome. Great time.
We had a meeting with my son in a college advisor. He's got to pick colleges to apply to. And I was thinking of Florida, Gainesville, and I said, nah, it's too hot there.
No.
One of the things he told the guy, he doesn't want to go anywhere where it's hot.
It's not hot it's only hot in the summertime when you're not in school. Come on.
Is it a good school, Rich?
It's a great
school. It is a good school.
That's a great school.
Yeah.
Great school.
All right. I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We will be back next week. Another TWiV is viral.